Hello, good evening. So this is Krishnaveni and thank you for joining me on today's lecture. So we are on the journey of the story of life on earth, and that's biology. So today we are going to talk about a very new chapter, that is the discoveries by the Austrian monks. So you would have guessed what it is, right? So that is nothing but your Mendelian genetics. So we are done with molecular genetics. So continuing on the topic of world of genetics, we are in the next section, that is your Mendelian genetics. So it's another huge and interesting aspect of your genetics. So no one can deny the fact that Mendelian genetics what forms the basis of genetics today. So all your genetic disorders that we are talking about today has been classified into two types. One is Mendelian disorders and the other one is non-Mendelian disorders. So what did this Austrian monk do? He was not even a scientist, he was a monk. So can you expect anything out of a monk? So according to us, a monk is a saint person who sits on a serene environment and who meditates, right? But imagine this monk did so much in the field of science, especially genetics, and he gave us so much of information that today we are able to decipher each and everything properly, and it's precisely accurate as well. So that is genetics. So I welcome you all once again to the world of genetics. Today we're going to talk about the Mendelian genetics. So Mendelian genetics is again an ocean, so it's a vast volume. So I'm sure you and I cannot finish Mendelian genetics in one lecture. So we will go forward with 3 lectures of Mendelian genetics, but still it is way less. So Mendelian genetics as such would take a month or 2 to finish. So it's a huge volume as such, and we are going to compress it further. Just like how your DNA is being compressed and compacted inside your cell, we are going to compress, and I'm just going to give you an overview of the entire Mendelian genetics. And anyways, for your NCERT level or for your NEET level, or in case of any medical exam, you will need it only in this level. You won't go much into detail, but you should know what causes your Mendelian disorders, because you're going to be the doctors of tomorrow, right? Okay, so welcome once again. So going forward, so here, this is your coupon code, so it's KR10. So please use this code to buy any product or any course with 10% off on the eCareerPoint app. So you can as well find my PPTs for your live class in this eCareerPoint app. So now quickly download your app. So maybe after the class, now don't just lose focus. So take our phone. So actually you can start the download and then listen to class. So by the time the lecture is over, you can log into the app. Fine. So make sure you use this coupon code. It's valid till 30th November only. Fine. So going forward, so I've already spoken about what is genetics, so why we have to study genetics. So it's a quick recap to all those who are new to my lecture today. So we have already seen the introduction of genetics, the classification of genetics when we were talking about the molecular genetics. But still, for all those who have joined me today, I will definitely not disappoint you, and I will still repeat the same thing, because so the more you listen to something, the more familiar you become. Okay, so genetics is not something you understand overnight. Okay, so genetics is not something that was discovered overnight. So it's like you need to comprehend, you need to relax, you need to understand each and every word with a lot of depth, because it's so complicated, but still it's happening. Okay, so that is the miraculous part here. So what is hereditary? So hereditary is the transmission of characters, resemblance as well as differences, okay, from the parental generation to the offspring. You resemble your father or mother, right? Or sometimes a child resembles his grandparents. But how is it possible? So what is being passed on? Sometimes you might have curly hair, but both your parents do not have curly hair, but your grandparents have, but say your grandmother has curly hair. So what is that information that is being passed on from the parent, from the parental generation to the offspring? So what is that information and how it is being passed on? So how do you understand that? Aren't you curious about that? So that is what genetics is about, right? So how do you look alike? But at the same time, we are not clones, right? We have differences, so we are not exactly similar. If you become exactly similar, that is identical, and we all become clones. Clones are like what is called identical copies. So we are not Xerox copy of each other's. We have our own differences. That is why we are unique. So what is that? So that caused the basis, that caused the curiosity, that rekindled the interest in so many scientists, and that rekindled the interest in me to study genetics as well. Okay, so that is what is hereditary. So before we actually go into genetics, it is important for you to understand these terms. So going forward, now what is genetics? So let me define genetics for you. So study of heredity, okay, variations and environmental factors responsible for these is called genetics. So genetics is a broad term where you will study hereditary. So hereditary basically means the transmission of characters, your differences as well as your similarities. So you're going to study that, variations and the environmental factors that plays a role in affecting these factors. Fine, so that is called as genetics. So, excuse me. So, who coined the term genetics? So, it is one of our scientists. So, our scientists are players here because biology is a story of life on earth. So, each lesson is a story and each player is your scientist. Okay, so heroes are your scientists and definitely your superhero is your DNA itself or the cell itself. Fine. Okay, so the person who coined the term genetics is William Bateson. So this is how it's, right? So you get one copy from your parents, sorry, one copy from your father and one copy from your mother. So he was Mr. William Bateson who coined the term genetics for us. Okay. So going forward, I will give you the classification of genetics. So genetics is a broad term. So we are going to talk today about Mendelian genetics. So how it is classified and what basis. So first is classical genetics. So classical genetics, classical is the oldest or the most highlighted one in case of genetics. So here we are going to talk about Mendel's principles, sex determination, sex linkage and cytogenetics. So cyto is cell, so study of cell genetics is called as cytogenetics. So that is our classical genetics. So classical genetics is also known as Mendelian genetics. So classical genetics is also known as Mendelian genetics. So the next one is your molecular genetics. So this is what we have been talking about in the last 4-5 lectures, and today we have started off with a new topic. So molecular genetics is the study of genetic material, structure, replication and expression. So here where we spoke about your central dogma of life, and then we spoke about each of the process of central dogma, that is replication, transcription and translation, right? So that was the molecular world of genetics. So it is followed by evolutionary genetics. So evolutionary genetics is a study of mechanism of evolution of genes, how the genes migrate, how there's a change in the gene frequency. So it's basically called as the population genetics. So just like your classical genetics is also known as Mendelian genetics, your evolutionary genetics is known as population genetics. For example, an Indian moves to America and he settles there. So there is a change in the gene frequency. So when an Indian migrates to America and he settles down and he has 4 generations in the American population, then there's a change in the gene population. So we will talk about population genetics as well. So as of now, we are done with the molecular genetics. Now we have moved on into classical genetics or Mendelian genetics. Fine? Okay. So going further, what shall we discuss today? So definitely we are not going to finish Mendelian genetics in one lecture. It's not possible for me to complete so quickly and it's not possible for you to understand. So we'll just go in our own pace. We will look into each of the topics in detail, understand them peacefully. So if you don't understand, make sure you put it in the chat box and I'm ready to teach again. So the beauty of genetics is understood only when it is, the beauty of genetics is actually understood only when it is taught properly. So though I think I teach well, only when you understand, that's where you can analyze its beauty. So make sure if you don't understand, you put in efforts to understand. Okay? So we'll quickly talk about Mendel's experiment. What did this Austrian monk actually do? So inheritance of one gene. So Mendel's laws, so Mendel basically hypothesized or proposed few laws. So we are going to see the first 2 laws and then we're going to talk about the concept of incomplete dominance, what is codominance, then inheritance of 2 genes and then law 3, that is independent assortment. Fine, so we'll end here with these 3 laws and certain concepts, but this will itself take more than 1 hour, but let's see how quickly we can finish. Fine? Okay. So first, welcome to classical genetics and it's basically the Mendelian genetics. So we are going to talk about the Mendel's principle. But who is this monk? Can you imagine him doing experiments like this? Okay, so Mendel, he is known as Gregor Mendel. So he was born in a very poor family. So basically they were farmers. So he had a lot of siblings. So he had an elder sister, younger sister, etc. So during a particular time in their life, they were in a very desperate state, like they didn't have money to support his educational needs. So it was very difficult for him to study. So then later he went on to do his farming in his field and everything was sorted. And finally he ended up in this in this in this you can say church. So he joined as a monk because they promised him if he joined as a monk, his educational finances or his educational aid will be taken care of. He was so interested in studying that he became a monk in the church, and that church housed a very beautiful garden, and since he's from a farmer family background, it was very intriguing for him. So he later continued his research, and hence today what we have is the Mendelian genetics. Okay, so you should be actually thankful to the church. You should be actually thankful to the garden in the church, and you should be thankful to the situations in Mendel life that he ended in this church, so that today we have what is called as Mendelian genetics. So aren't we not being selfish for a moment? Just because we need to understand, we are actually being blessing his disguised life, right? So that should not be the case, but it's just on a fun note. Okay, so going forward, so he's known as Gregor John Mendel. Okay, he's known as the father of genetics. So without him, the word genetics, actually what it means could not be defined today. Okay, so he is known as the father of genetics, and Mendel did a statistical study. So basically he took out biology and mathematics and combined them both. Okay, for a few years it astounded so many people. So many people could not come into terms to agree that biology is combined with mathematics, and hence he was a failure at his time. But later when his work was rediscovered, today we know how amazing his work is, right? Okay, so what did he do? So I give us so much of hype, right? So Mendelian genetics, he was a monk, but still he did so much, etc., etc. But what did he actually do? He did this. He discovered that individual traits inherited as discrete factors which retain the physical identity in a hybrid. For example, the example that I gave you moments ago. For example, your grandmother has curly hair and you have the same curly hair. So what is that being passed on? So there are some discrete factors which are being inherited from that generation to you. So that is what is showing its physical impact, right? So that is what Mendel also found out. So these factors, he named them as discrete factors, but later these discrete factors are now known as genes. Fine? So how do you define gene? So in case of molecular biology, we define it in terms of molecular biology. We define it in terms of molecular genetics. That gene is nothing but a sequence on your polypeptide that codes for a protein. So now coming to classical genetics, we'll define it as a unit of heredity that may influence the outcome of an organism's trait. Okay, so you might get confused as to what is this trait. Don't worry, we still have more to go. I will explain to you what is trait. Fine, so this is about Mendel. So he did his work. You might have heard this n number of times. So he performed his experiment on pea plant, and the scientific name for your pea plant is Pisum sativum. Fine. So how do you write a scientific name? You either italize them. Okay, so since it can be done in computer, I have italicized them. So otherwise, when you're writing it, first letter is capital, starting of the second letter is small, and then you underline both separately. So Pisum sativum, so that is how you write. So it is basically a pea plant, the normal garden pea plant. Fine. So you would have seen these pea plants, right? So he performed his experiments on these pea plants. So that is called as Pisum sativum. Fine. But why did he choose these pea plants? Just because the pea plants were present in the church that he was living in? No, but basically there were other reasons as to why he chose the pea plant. So the first reason is that it had well-defined discrete characteristic. So each characteristic of the pea plant was very well discrete. For example, the flower was discrete, the fruit was discrete, the seed was discrete, the leaves were discrete, the branch were discrete, meaning they were different. Nothing was connected and it was easy for him to study. And that's why he chose. So that is one reason. The second reason is bisexual flowers. So bisexual, so bi is 2, it means it has both the male and female flowers. So when he had to do hybridization or when he had to do pollination, it was easy for him. Okay. Then predominant fertilization. Okay. Excuse me. The fourth one, easy hybridization. And fifth one, it was easy to cultivate and relatively short life cycle. So you didn't need much of fertilizers to grow these pea plants, so it was easy for him to cultivate. He didn't need a lot of support, he didn't need a lot of labor to do it, and it had relatively a short life cycle, meaning it was not an annual crop, it was not a perennial one, but it was a plant with relatively a short life cycle. Fine, so this is the reason he chose your pea plant. So going forward, so what did he study in the pea plant? So we understood Mendel did experiments and he came up with the terms that there are discrete factors that are passed from one generation to the other. Fine. And he studied in the pea plant. So we saw the reasons why he selected the pea plant. But now, what did he study in the pea plant? So that is what we are going to see. So he studied these 7 characters. So characters of an organism are described as characters or traits. So for example, if I say height is a character, height of the plant is a character. Okay, so that is called characters or traits. So traits Mendel studied were very discrete and clear. So not any 2 traits are not related. Fine. So he studied these 7 traits in a pea plant. So seed shape, seed color, pod shape, pod color, flower location, and plant size. So each of these are 7 characters. So each character has 2 options and this is called trait. For example, when I say height is a character, tall and dwarf are the 2 options possible in height. So these 2 are called as traits. Fine. So this is called as characters. And the 2 opposite variations or the 2 options present in a character is called as traits. Fine, is this understood? Okay. So known as Mendelian characters. So Mendel studied the following 7 traits. Fine. So next term that you need to understand is alleles. So what are alleles? So genes which code for a pair of contrasting traits, slightly different forms of a same gene. For example, I told you height is a character, right? So it has 2 traits. One is tall, one is dwarf, right? So there should be a gene that codes for tall and dwarf, right? So this is the character and these are the 2 traits, tall and dwarf. So there should be a gene that codes for tall and there should a gene that codes for dwarf, right? So these 2 genes that codes for the varying or the opposite trait is known as alleles. So they are the same gene that is the gene that codes for height. So in the gene that codes for height, there are 2 traits. One is tall, one is dwarf. So the genes that code for tall and dwarf, they both are contrasting or opposite characters. Hence we call them as alleles. Okay. So genes which code for a pair of contrasting characters, so slightly different forms of the same gene. Fine. Okay. Now we are going to talk about wild type versus mutant allele. So wild type is the prevalent alleles in a population, which means the one which are present naturally in a population, they are wild type. So they have to be the majority. So then you call them as wild type. So wild type, you represent them as WT. Now the other counterpart is mutant alleles. So which means they are not present like wild type. So there is some mutation, there is some change in the mutant alleles. Okay, so they are called as the mutant ones. So they are less than 1% in a population and they are altered by mutation. So what do you mean by mutation? So mutation is a change in the genetic sequence. Fine? Okay. So the next term, so the first term that I want to talk to you about was alleles. So the second term is wild type and mutant alleles. The third one is dominant and recessive. So literally from the term dominant, you understand what it means, right? It has more power over the other. So that is dominant. So you can say my brother is always dominant to me. So he always bosses over you. But you are very recessive, meaning you accept whatever he does. So you give him that position, right? So that is called as dominant. So dominant allele masks or hide the expression of recessive alleles and it is always represent in the uppercase. For example, the same example, here I said capital T stands for tallness. So tallness is dominant. Okay, so when it is dominant, I write it as capital T, capital T. Dwarfness is recessive, so I write it as small t. Fine. So recessive alleles exerts its effect only in homozygous condition. Homozygous condition means only when there are two same type of alleles, they exert their effect. Otherwise, when you have a condition like this, your capital T is dominant over small t. So obviously your capital T will be expressed and not the small t. Okay, and they are masked by dominant allele in heterozygous condition. So this capital T and small t is your heterozygous condition. Fine. So your recessive ones are always represented in your heterozygous condition. So that is in your lower case. So just like this picture says, your dominant ones are more bully type. So they have more strength, they have more energy. So that is why they are dominant. Okay. So this is how it is. So you have homologous chromosome. So I guess this is clear. This is how a homologous chromosome looks like. So you have a gene here. So this is A, and you have another gene here, that is small a. So this is the dominant allele and this is the recessive allele. So this is how they are arranged on your chromosome. Fine. So going forward, the next term that I want to discuss is homozygous and heterozygous. So we have already discussed what is homozygous. Homo means same, hetero means different. So when I say TT, it is homozygous. Or when I write small t, again it's homozygous. But when I write capital T, small t, it is heterozygous. So this is what essentially I am going to define for you. So each parent, that is diploid parent, has two alleles for a trait. For example, we also have the gene height. Okay. So say this is the gene for height and we have two alleles here. So capital T, small t. Right. So this is how it is. So a diploid cell has two alleles for a trait. Okay. So for example, this is your dominant homozygous, this is your recessive homozygous. Fine. So now defining homozygous for you, it possesses two identical alleles for a trait. For example, when I say TT, capital T, so both the alleles are identical, right? So both are lowercase, both are recessive and both are dominant. So this condition is called as homozygous. So now what is heterozygous? Okay, so before we go into heterozygous, we have what is called as homozygous dominant. So when I write two dominant alleles together, it is called homozygous dominant. So that is two dominant alleles for a trait. So now this will code for the tallness of a plant. Fine. So the next one is homozygous recessive. That is when I write two recessive alleles together. So this will code for dwarfness. So this is recessive. So recessive alleles will be expressed only when they are in homozygous condition. Right? So now moving on to heterozygous. So heterozygous genotype, it possesses one of each allele. So it is simply this, capital T, small t. So here which one will be expressed? Your tall one, because it is the dominant allele. Fine? Okay. So here I have a picture. So both are the same. So it is homozygous. So it can be either AA or it can be small a. Again, it's homozygous. It can be dominant or recessive, right? The next one is heterozygous. It can be capital A and small a. Okay. Now, going forward, inheritance of one gene. So Mendel gave us this concept of inheritance of discrete factors and we later call them as genes. But does one gene get inherited or two genes get inherited? So that is this experiment. So you would have seen this experiment a million times. So Mendel basically worked with a pea plant. So what he did with a pea plant? So the pea plant, again, he took the character here as height. Okay, so the character that we are talking about is height here. So he initially took a tall pea plant and he took a short pea plant. So we call them as dwarf pea plant and we are going to pollinate them. So we are going to cross pollinate them. So this is the parental generation. So your parental generation has a tall plant and a dwarf plant. Fine. So you call this as P or parental. Fine. So when you cross these two, so what is your first generation offspring? So this is the parental generation. So whatever their offspring is, you call it as F1 generation or the first generation or the filial generation. Fine. So when you cross these two, you get two tall plants according to Mendel's experiments. Now, so both are tall plants. So you cross this with another tall plant, you cross this with another tall plant. So basically, you take a tall plant, you take a dwarf plant, okay, and then you cross them. So what you get is a tall plant. Now you cross this tall plant with another tall plant. So what is your final F2 generation? You have 3 tall plants and 1 dwarf plant. Wait, let me show you. So F1 generation resembled one of the parent. So this is your F1 generation. This is not your F1 generation. Only one offspring you have in F1 generation. That is tall plant. So now for this F1 generation, you are pollinating it with another tall plant from some other background. Okay, but it's a tall plant. So when you cross them, you have F2 generation. So which I mean to say, in F1 generation, the offspring is similar to one of the parents. Here it is similar to this parent, right? Fine. In F2 generation, they are identical to both the parental type. So see, you have tall, tall, tall and dwarf. So they are identical to both the parental types. But here the point to understand or the point to note is that there is no intermediate type. Meaning there is no plant with a medium height. So they are either tall or dwarf. They don't have an intermediate height, right? So Mendel said that something is being stably passed down unchanged from parent to offspring through successive generation. Though in F1 generation you do not have dwarf plants, it does not mean that the dwarf trait is forgotten by the plant. See, in F2 generation you have a dwarf plant. So in the immediate parental generation you do not have a dwarf plant, but still it remembers the information that was present here, right? So now we'll write this in the form of a cross so that you will understand. So since you are talking only about one type of character here, these are called as monohybrids. So these factors were called as genes. Okay, so genes control only one character here, because here the only character that we're talking about is height. So your gene is controlling your height and hence you have only one character, so it is called as a monohybrid. So mono is one, right? So such a cross is called as a monohybrid cross. So let's quickly write the cross. So parental generation, so that is my P generation. So you have a tall plant. So I will write capital T, okay. So the other plant that is crossed is dwarf plant. So it is small t. So I take only one gamete from here. So gametes are always haploid, right? So now this will give me, if you have a plant, if you have a heterozygous allele like this, your capital T is dominant over your small t, hence your plant is tall. So this was your F1 generation, right? Now with this you are going to self it. You are going to actually, self means identical plant. So this was your F1 generation product. You will cross this with an identical one. So now what will be your gametes? So from this one gamete is capital T, from this one gamete is small t. So capital T, small t. So now you will multiply and write. So obviously this is tall, this is tall, this is tall, but this is dwarf, right? So in F1 generation you have all three combinations, and in F2 generation you have both the parental combinations, that is tall and dwarf, but in F1 generation you have only tall plants. Fine, so that is understood here. So these are the offsprings of your F2 generation. Okay. Okay. So this is the cross that I was talking about. So it has TT. So you gametes have only one allele coming out. Okay. So this is your F1 generation. So then you again self it with another one. So this is how you find out the gametes. Okay. So recessive parental trait is expressed without blending in the F2 generation. Though here you do not have a dwarf plant, you have both the tall plants, still the dwarfness characteristic is being expressed in the F2 generation. Okay, so that is what it means to say. So what do we conclude from that point? That gametes produced by meiosis. So by now we will talk about cell cycle in our class 11 3 days as well. So there I will teach you about meiosis and mitosis. So we will understand meiosis produces gametes. Okay, so the alleles of parental pair, they separate or segregate from each other. So understand, if you have an allele like this, okay, so this will separate independently of this will separate independently of this. Fine. So that was the conclusion of Mendel. So what is this representation of this box called as? So we are writing down the gametes and then we are multiplying it to find the generation, right? So we are trying to find the phenotype. So this is called as Punnett square. It is the graphical representation to calculate the probability of all genotypes of offsprings. So what do you mean by genotypes? So genotypes is a genetic makeup of an organism. Okay, so phenotype is the physical observation. So here the phenotype is tall plant, tall plant, tall plant, and this is a dwarf plant. But genotypically, how do you express? It has TT, it is dominant. Okay, so it is homozygous dominant, this is heterozygous, and this is homozygous recessive. So this last one that I was talking about is the genotype. Fine. So now you have what is called as a test cross. So this is another term that I am introducing to you. So what is a test cross? So when the F1 progeny is crossed with a recessive parent, for example, this is the F1 progeny. When you cross this with a recessive parent, you get 50% dominant and 50% recessive. So what do you understand? So this is the F1 parent, so which says F1 progeny. So my F1 progeny is TT, so I am crossing it with a recessive one. So when I cross, make a Punnett square like this and tell me whether you get 50% dominant and 50% recessive. Okay, so these were the conclusions of Mendel. Apart from text cross, you have a back cross. So when the F1 individuals are crossed with either of their parents, okay, it is done to find to whom they are genetically similar to. So test cross, when you cross the F1 generation with a recessive alone, okay, so that is a test cross. But what is a back cross? When you cross the F1 generation with either of the parent to find how close they are to each parent. For example, some people say you are more like your mother than like your father. So how close you are to your parents? So that is explained by your back cross. Fine? Is this understood? Okay. So going forward, so this is the difference between back cross and test cross. So understand this. So you have an unknown dominant phenotype and you have a known recessive one. Okay. So now it can be either BB or it can either be B or B. It can be homozygous or it can be heterozygous, right? But here it is understood it is homozygous recessive. So you cross this with first dominant one and then get the phenotype. Again you get the phenotype. So when it is done with the recessive one, you get 50% of both the ones, dominant and recessive. So this is called as your test cross. But when you do it with both your parents, then it is called as a back cross. Fine. So here I have an example of a back cross for you. So the phenotype of parents are pure tall. So your plants are tall here and here they are dwarf. So the genotype is TT and TT. So you take out the gametes. So your F1 hybrid is capital T, small t. So when you make this with the dominant one, it can be either dominant or the recessive one, and then you see to which parent they are very close to. Fine? Okay. So going forward, the first law of Mendel was the law of inheritance. Okay, so sorry, the cumulative or the collective term of Mendel's laws were called as the law of inheritance, and the first law is the law of dominance. So what does the law say? When parents with pure contrasting characters are crossed together, only one form of trait appears in the next generation. So the hybrid offsprings will exhibit only the dominant trait in the phenotype. For example, in the parental generation, that is of monohybrid cross that I am talking about, you had this, right? Sorry, this is capital T, this is small t. So this was your offspring, right? So he means to say in the F1 characteristic, it will resemble only one of the parent, that is with the one that is being dominant. Okay, so that is called as a law of dominance. So he means to say the F1 generation will have a phenotype that will resemble the phenotype of the dominant parent. Fine. So each character is controlled by distinct factors that occurs in pairs. So if pairs are heterozygous, one will always dominate the other. For example, this is heterozygous, so your capital T is dominating over your small t, right? In F1 generation, only one parental phenotype is expressed, that is the tallness. In F2 generation, you have both the parental alleles, that is you have both tall and dwarf plants in the ratio 3:1. So you saw figures like this, right? So after the Punnett square, you had all these possibilities. So 3:1 because these three are tall and this was dwarf. So this is 3:1. So remember, recessive traits are always marked by your dominant traits. Fine? So now talking about the second law of Mendel, that is law of segregation. So what does this law state? So during the formation of gamete, okay, so this also we have discussed, but putting it in terms of the law. So during the formation of gamete, each gene variant separates from each other so that only one allele will be present in each gamete. For example, if the parent has TT and small t, so the gamete will have only one allele. So this separation is independent. So this is called as the law of segregation. Fine, is this understood? For example, this is the female parent, so the gametes are T capital T. So male parent T capital T. So this separation of gametes is called as the law of segregation. Fine. So in F2 generation, both parental alleles are expressed without blending. So in F2 alleles, you have both tall and dwarf characteristics. Right? So this law is based on 4 concepts. So what are the concepts? So gene exists in more than one form of an allele. So they are always in pairs. So when gametes are produced by meiosis, allelic pairs separate, leaving only one gamete with one allele. So see, there is only one gamete with one allele, right? So every organism inherits 2 alleles for each trait. So for the character height, you have 2 traits, that is tall and dwarf. So you get one allele from your father and one allele from your mother, right? So 2 alleles of a pair are different. So one is dominant and other is recessive, at least here in this case. Now you have another term, what is called as incomplete dominance. So you understand dominance, that is being expressed. Now you understand recessive, that is being masked. Now what is this incomplete dominance? So experiments with other plants using different traits was performed. Now we are done with the character height. Now we have moved on to a next character. So it was found that your offspring resembles neither F1 or F2, it neither resembles both your parental type, but you have an intermediate type. So that is called incomplete dominance. Example is the inheritance of flower color in your dog flower. So that is what we are going to see. So such pretty flowers, right? So no one ever knows this is called as dog flowers. But they look really pretty and they really look colorful. So it gives you a sense of peace, right? Okay. Now here we have the cross. What is incomplete dominance? You have a red flower plant and you have a white flower plant. When you cross them, you get a pink flower. So it neither resembles either of your parents, right? So that is why it's called as incomplete dominance. So but when you self this with another pink flower, what you get is a combination. So that is the beauty of this. So you have a red flower, you have a white flower. So the red flower is represented by CR, CR. The white flower is represented by CW, CW. So you have one gamete here, one gamete here. So the F1 generation is CR, CW. So this is a pink flower. Now when you cross this with another pink flower plant, so you make a Punnett square and you find that you have one red, one white and 2 pink. So there is an intermediate color that is possible. Hence it is called as incomplete dominance. So phenotypic ratio, so phenotype is the physical observation. So red is to pink is to white. So 1:2:1. Fine. Genotypic ratio, so this is basically CR, 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 CW, CR, CW, CW, CW. So these two codes for pink. So 1:2:1. So your phenotypic ratio and your genotypic ratio both are the same in case of incomplete dominance. Next is the term codominance. So what does codominance mean? So F1 generation resembles both the parents. Okay. In case of Mendel's monohybrid cross, F1 generation did not resemble both the parents. It only resembled one of the parent. So in case of codominance, your F1 generation will resemble both of your parents. So one example that we are going to talk today is the different types of RBCs that examine your ABO blood grouping in humans. So you would have also heard about this. So we all have A blood group, B group, O group, AB, O positive, O negative, right? So this ABO blood grouping is controlled by a gene called I. Okay, so basically a gene will have two forms, that is called alleles, but here it has three alleles. It's called IA, IB and I. So when is written in lowercase, it means it is recessive, right? So that is how you denote recessiveness, and that is how you denote dominant characteristic. So IA, IB seems to be dominant because that is how it is represented here. So it produces a slightly different form of sugar, so it is dominant over I. Both your IA and IB are dominant over your small I. Okay, so the allele I, it does not produce any sugar. Fine. So when there are two alleles, more combinations are possible. Now there are three alleles, so 6 different combinations are possible. So ABO blood grouping is a very good example of multiple alleles found when population studies are made. So you will understand the significance of this line when we are talking about population genetics. So now this is actually your ABO blood grouping. So if you have IA, okay, so if you get this from your mother and you get this from your father. So from your mother you get IA and from your father also you get IA. So both IA is as such dominant. So it is homozygous dominant, so you will have A blood group. Fine, no doubt? Okay. So you get IA from mother and IB from father. Anyway, this is also heterozygous dominant, so both are dominant, IA, IB, so you become AB. Now, your mother gives you IA and your father gives you a recessive gene, which means IA is dominant, so you will get A group. Now, IB is given by your mother, IA by your father. So it is IA, IB, so both are dominant, so again you have AB group. Okay, so your mother has AB, your father also has AB, sorry, BB, so hence your blood group is B because B sugar is produced. Your mother has B, your father has I, so obviously I is recessive, so you will have B group. What if both of them have a recessive allele? That is both of them have I and I, small I, which means you will have O group. So this is called codominance. When your F1 generation resembles both the parents. So when you look in the case of AB, so everywhere it resembles both the parents. So in case of AB and AB, it is very visible because they are distinct alleles, right? So here you have 6 combinations possible. So going forward, codominance. More example of codominance. So occasionally what happens is one gene produces more than one effect. So sometimes people are so smart, right? You find the student who can do 2 things brilliantly, but at the same time we are struggling to do one thing positively better. But there are some occasional cases where one gene produces more than one effect. For example, I'll give you a small example here. You have starch synthesis in pea seeds, okay? So it is controlled by one gene. So what happens is there are 2 alleles, BB and B. So capital B, that is homozygous dominant, has an efficient synthesis and it produces large starch grains, okay? Small b, that is homozygous recessive, it is less efficient and it produces small starch grains, right? Okay. After maturation, so they are basically pea seeds. So after maturation, BB produces round seeds, small ones, the recessive ones produces wrinkle seeds, right? But so this is round, this is wrinkle. So the heterozygous one will be round, right? Because B is dominant. But once after maturation, this produces an intermediate size which is neither small nor big. So this shows incomplete dominance. Thus, dominance depends on the gene product and the product of a particular phenotype. So if you look here, so initially it produces big granules and then small granules. But after maturation, it produces what is called as the intermediate size. So one gene can affect more than one character. Now, coming to inheritance of 2 genes. So long it was very smooth. We were talking about only one character, so no headache. Now when you combine 2 characters, what will happen? So pea plants with 2 different characteristics was crossed, so it gets complicated. So what are the 2 characteristics? So yellow color and round shaped seed. So you're talking about the color and the shape here, okay? So yellow color, round shape versus green color and wrinkled shape. So when you're having 2 characters, you call them as dihybrid crosses because di is 2. So they are differing by 2 traits. So you have a yellow color and round. So capital Y, capital R. So green wrinkle, small y, small r. So it is understood this is recessive. So when you cross them, you have two different characteristics. So you will have two different types of gametes here, right? So when you cross, it will become capital Y, capital R, small r. So it becomes yellow round because the dominant ones Y and R are present. Fine. But when you self this, so that is the procedure of crosses, right? So when you self this with another, what will happen? So you will write down your gametes here, and again you will do your Punnett square crossing. So what are the gametes? It can be capital Y, capital R, right? It can be capital Y, small r. Now it can be small y, capital R, small y, small r. So when it is done selfing, you will have the same gametes here, and then you multiply and write. So looking at the figure, what can you make out? Here the dihybrid ratio is pretty large. So when you look at the phenotypic ratio, it is 9:3:1. So phenotypic ratio is the physical observation. So how many yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? So it is 9 round yellow. How many green? 1, 2, 3, 4. Sorry, 1, 2, 3. So you have 3. So 9:3:1. So you have 3 yellow wrinkle and you have only 1 recessive one, that is your green wrinkle. So this is the phenotypic ratio, 9:3:1. So 9, you count the yellow color, the dominant yellow ones. So 3 is this yellow. So 3 is this yellow, which is yellow but wrinkled. 1, 2, 3. So these 3 are similar, right? So the other 3 is fully green. Okay. The last one, 9:3:1. So 1 denotes the recessive one, that is your green wrinkled. Fine? Okay. Now comes the third law, that is law of independent assortment. So what does this law have to say? When two pairs of traits are combined, like your color and shape, when they are combined in a dihybrid, segregation of one pair of character is independent of the other pair. For example, you had something like this, right? So from this you take one gamete, from this you take one gamete. So the segregation of this is independent of each other. So that is called as your law of independent assortment. Fine. So this was the third law under the law of inheritance. Fine. So in a nutshell, what did we discuss? I think this was pretty saturating because we spoke about both your monohybrid cross and dihybrid cross as well. So quickly summarizing what we spoke for an hour. So genes are the ones that are stably being passed on from one generation to other, and Mendel called them as discrete factors, which are later known as genes. So the allelic pairs separate independent of each other. You have what is called as dominant and recessive alleles. Then we spoke about the law of independent assortment, law of dominance, and law of segregation, right? Okay, so everything is hereditary. So why do I have long face? Why do I have curly hair? Everything is hereditary. Fine. Now, since we spoke so much, so let's quickly have a few questions so that we stay updated. So the first one, dihybrid cross proves the law of. So just a moments back, just a few seconds back, we spoke about dihybrid cross. So dihybrid cross is involving two traits, that was your round color and your yellow and round shape, round seed, and then green and wrinkle seed, right? So it proves the law of segregation. Fine. So the two pairs segregate independently of each other. So how many types and in what ratio the gametes are produced in a dihybrid cross? So this is your dihybrid cross, right? So you do a selfing. So you have 4 types of gametes being produced. So what are the gametes? It can be capital Y, small r. Sorry, capital Y, capital R, capital Y, small y, capital R. So you have 4 types of gametes which are produced in the ratio 1:1:1, right? So that is the answer. So third question, Mendelian ratio 9:3:1 is due to the law of segregation. So that is your dihybrid cross. Fine. So the last question. So this is the genotype. So in genetics, the problem is you will have questions like this. Okay, so let me solve it elsewhere. So in genetics, you will have questions like this. They will give you the genotype and ask you the gametes that are formed, the number of gametes that are formed. For example, here the question is, okay, I will solve it here. So this is the small c, right? So how many gametes? So this is a tri-hybrid cross. So here it is a homozygous one, but these are heterozygous. So how do you find out? Right? Small c, capital C, small c. So 1, 2, 3. So this is 1, 2, 3, 4. So you have 4 types of gametes that are being formed. Fine. So this is how you solve questions like this in genetics. So you have 4 types of gametes that are being formed. Fine. So with this, we come to a happy ending of the first lecture of your genetics. Fine. So thank you for joining me in this session, but join me for so that we complete the entire story of Mendelian genetics, and we'll talk more about what your genes actually do. So you have more about a chromosomal theory of inheritance, crossing over, linkage. So they give us, they make us land in a conclusion about our Mendelian disorders. That is way more interesting. So make sure you join me for more such lectures. So if you like the session, do like, share and subscribe. So this is me signing off. Until then, thank you.